Hello, in this video we'll talk about the polymerase chain reaction or PCR. Within these 10 minutes, this concept will be totally clear to you. This video is dedicated for school students because in school, generally biology is taught in a boring fashion. You are trained to mug up information and things are not really interactive. So in this video, I'll be really describing the concepts of NCERT books in an engaging animated fashion. So stay tuned till the end. This concept is taken from chapter 11 NCERT textbook of class 12 and the chapter name is Biotechnology Principles and Processes. Polymerase chain reaction is a method widely used in molecular biology labs to make many copies of DNA segments. So it's like a molecular Xerox machine. Imagine this is a cell and inside the nucleus of the cell there would be different genes and this is one particular gene. And out of many gene segments we want to amplify this particular portion. And one way we can achieve that is by using PCR, which allows us to amplify a specific segment and make thousands of copies of that particular segment only. And we decide which segment we want to amplify. And that's the biggest advantage of this technique. So polymerase chain reaction works like a Xerox machine. It's kind of making copy of DNA, right? Exactly. In a Xerox machine, you have to copy from this page to that page and you get the desired copy, right? And in this PCR reaction, using specific instruction, you tell the machine that you have to copy from this region to that region. And exactly at the end, you get that particular region of interest. So both are quite similar. And this is the crux of the story. Before delving into details, let me tell you, if you are worried about NEET UG, Unacademy, which is India's biggest online learning platform, get you covered. They have lectures, notes, doubt clearing sessions, which would help your journey really smooth. Right now, if you're thinking about NEET UG 2023, one batch is starting right now, which is known as Enthuse batch. This batch is starting from March 23rd, 2022. If you have any query regarding this batch, you can call in this given number. All the enrollment link is provided in the description. Make sure you check the description. In this batch, you would be taught by the popular educators all across India who would prepare you throughout this journey and make your journey a bit more enjoyable, easier, and make you prepared for the final exam. Now, Unacademy is also launching a Ask a Doubt platform where if you have any doubt regarding any concept, any particular question, or anything, you can post it on the platform and educators would actually get back to you with a solution of that doubt. This platform is amazing for school students. Do check it out. Now the prices would be hiked very soon. So if you are thinking of taking a subscription, thinking of taking it really seriously, take a subscription within March 30th, 2022. Otherwise, you would face a price hike. Anyway, let's hit a virtual lab to understand the process of PCR, how exactly it is done. So PCR is generally done in a small tube like this. And in this tube, you add several reagents. All of these reagents are generally liquids. So you add template DNA, maybe you have extracted or purified DNA. You add that template DNA. You add forward and a reverse primer which binds to specific segment of the DNA. You add dNTPs which are the building blocks of these DNA. You add Taq polymerase which is isolated from a particular extremophile known as Thermus aquaticus. So this is a bacterial derived polymerase which can withstand a very high temperature. Lastly, you add Taq buffer, which is an optimal buffer for this Taq polymerase to work. Generally, it contains magnesium chloride, and magnesium ions are super important for this polymerase function. All of these components comes as a liquid, and you have to add all these liquids, and you have to add microliters of these liquids inside this particular tube. and thus you create this reaction mixture. After that, you put this reaction mixture into a PCR machine. This PCR machine is otherwise known as thermal cycler. Why this name? Because this machine can change temperature very rapidly. The way you program it, it would change the temperature. So let's see what really happens in the PCR machine and how the program is kind of like set up. So the first step is denaturation. In this step, temperature rises to 95 degrees centigrade, and this step holds for like 3 minutes or so. In this particular step, the DNA strands would be separated from each other. As the temperature rises, the hydrogen bond between these DNA bases get dissociated, and thereby the strands separate. The next step would be primer annealing. The primers that you have provided in the reaction mixture would anneal at a temperature around 55 to 60 degrees centigrade. This temperature is variable for what type of primer we use and also the constituent of the primer. If there is a difference in GC content in the primer, there could be difference in the temperature. But anyway, this temperature is lower than 95 degrees centigrade. So when temperature drops down, primers can anneal to specific regions of the DNA based on base pairing. So the particular region where the primer is complementary to, it would bind to that particular region. Now from the 3' end of the primer, there would be extension with the help of the Taq polymerase, and Taq polymerase is operational at 72 degrees centigrade, which is quite high temperature. So Taq polymerase would extend these primers and create a copy, and in each cycle the number increases. For example, this is cycle 1, you have 2 copies after cycle 1, you would have 4 copies after cycle 2. That means it's always getting in power of 2, right? 2 to the power 2. Then in the third cycle you would have 2 to the power 3, that means 8 copies. So now you can understand it follows this kind of formula, nt equal to n0 2 to the power n. So initially there would be a lag phase, then there would be a log phase, and followed by that there would be a plateau. This is how exactly PCR reaction look like in terms of an equation. Maybe that equation is not important for NEET purposes, but it's good to know. Now you would get, at the end of the PCR reaction, you would get thousands of these kind of amplified copy. Now one way to visualize these reaction happen properly is by looking at the agarose gel electrophoresis. Here in an agarose gel you load the PCR products. Now you know this PCR product would amplify a particular segment of DNA with given amount of bases. So from the length you can get an idea, and this is a real gel how it looks like. So you can see this particular PCR product is slightly, it's around like 800 base pairs. So you can see a specific amplification has taken place. But look at this particular gel. Here you see there are multiple bands. You are expecting one band around 800 base pair, but why do you get multiple bands? This can be caused due to non-specific amplification. So this particular band encircled in yellow is your target band. Look at other wells. You see multiple spurious bands. This might be due to non-specific amplification. That means with your specific product, there is non-specific products as well. So this is one of the disadvantage of PCR reaction, that it can also give non-specific amplification, but just tweaking the parameters, changing the primers can give you specific amplifications. Now let me tell you quickly the applications of PCR. PCR is used in wide variety of fields. For example, biomedical research, medical diagnosis. You all know about RT-PCR that helps in diagnosis of COVID, right? If you want to learn more about RT-PCR, you can click on the video in the I button. Forensic purposes, genetic engineering, phylogenetic analysis, DNA sequencing. For many cases, PCR is used widely. So, do you know how PCR was discovered? It was discovered by Kary Mullis in 1993. And this discovery story is amazing, and it is basically discovered, and the idea was chalked out behind a Walmart bill. So one Walmart bill changed the entire molecular biology field. If you want to learn more about this story, click on the I button. So let me give you a quick idea how PCR can be used in a medical diagnosis. Let's say you are affected by a bacteria. Now from your sample, one can look for a particular gene which is very specific to that bacteria. I'm only giving you an overview. And if that particular bacteria is present, you might get that bacteria specific DNA in your sample. In this case, this green DNA or green amplicon is that bacteria specific amplicon. If you are not affected, you should not get a band corresponding to this. And if you are affected, you should get a band corresponding to this green band. So in this case, in this particular example, you can see these 5 lanes are from 5 different patients, and you can see patient 1 and 2 is affected by the particular bacteria, 3 and 4 are not. In 5, the bacteria might be affecting the patient as well. Now, I would end this video with an open question. You know, this polymerization and PCR reaction is pretty much similar to DNA replication. DNA replication is also another way by which the cell makes one extra copy of the DNA, right? But why does the cell stop with one copy, right? In PCR, you get multiple copies. But why does cell does replication only one time in one dividing cycle? Have you ever wondered about this thing? If you know this answer, let me know in the comment. Anyway, all the notes and flashcards would be available in my Facebook page. Links are provided in the description. I have bunch of dynamic flashcards which would help your preparation. As usual, like, share and subscribe. You can support my channel on Patreon. I'm also present in Unacademy, which is India's biggest online learning platform, using